Break is over. The second part of the lecture, causative agent of botulism. I did explain you about clinical forms of botulism and neopathogenesis of botulism. Botulism is a toxinemic infection. I'm going to remind you, what does it mean, toxinemic infection? This term is used in some cases in toxinemic infections. The pathogen, bacteria, remains at the entrance gate, and the pathogenesis of the diseases is mediated by the action of exotoxin. Exotoxins, exotoxin or maybe exotoxins are circulated in blood, and circulation of exotoxins in blood is named toxinemia. So toxinemia means circulation of exotoxins in blood. Bacteria don't circulate in the bloodstream. So in case of toxinemic infection, just exotoxins are circulated in blood. And I told you at the first part that for every exotoxin, there is some 13 organ of action or system. So in case of botulism, from the stomach or wound, botulinum toxin penetrates the blood vessels, and with blood flow, it enters various organs and tissues. It doesn't act, I'm going to remind you, it's not enterotoxin, in spite of very often it goes through digestive tract. It doesn't act on digestive tract. It's not enterotoxin. It is neurotoxin. So it affects mainly the nervous system. Peripheral nerve endings, it acts on synapses, and then alpha motor neurons of the spinal cord, which causes muscle paralysis. The reproduction of bacteria in the body most often does not occur. Sometimes bacteria, there are no live bacteria in this food, but exotoxin is accumulated. So toxinemic infection. I am going to remind you, toxinemic infections are infections caused by staphylococci. They are able to produce a lot of exotoxins. Then disease caused by Clostridia, tetanus, botulism, gas gangrene, and diphtheria, and whooping cough. There are toxinemic infections. Pay attention. The main pathogenesis factor is exotoxin, and the pathogenesis is connected with exotoxin action, and clinical symptoms appeared due to 13 exotoxin action. So clinical manifestations in case of botulism. The incubation period is usually 24 hours, but can vary from 6 hours to 96 hours or more. Sometimes it takes a few days, 10 days maybe even. It depends on the nature of the product, the amount of accumulated toxin, and the condition of the patient. First, disease symptoms. Students do mistake very often because they remind that botulism is food intoxication. So they are answering automatically that the patient has vomiting, nausea, vomiting, diarrhea. No, please pay attention. It is neurotoxin, so patient has no diarrhea, no. So gastrointestinal disorders are not regularly prominent. Just a few patients did notice abdominal pain, very rarely, sometimes nausea, then dry mouth or hypersalivation, symptoms of gastrointestinal disorders. But mainly, of course, disorders are due to action of the botulinum toxin on synapses. nerve paralytic. At first, patient is not able to see well, fog before the eyes, double vision, drooping eyelids, squint. Then, so at first patient are not possible to see well. Then muscle weakness, then hoarseness, then swallowing disorders. Patient is not able to swallow food, not possible to hear well. Then changes, the voice is changed, then lethargy and headache. So symptoms of destruction of the nerve system. There is photo of the patient with botulism. Drooping eyelids and squint. Patient is not able to open eyes. And then symptoms of botulism, dry mouth, do you see? Maybe hypersalivation, maybe dried mouth. And in case of wound botulism, the same symptoms, but this patient has wounds and the same symptoms at first, problems with the vision. Then newborn botulism. The same symptoms are developed, newborn botulism. So no tears and closed eyes. And then paralysis of the muscles of the neck, limbs, and respiratory muscles, and the heart muscle occurs, resulting in death. Mortality rate is very high, up to 60%. Immunity after a disease. Some patients are able to recover if the diagnosis is proved and immediately the treatment is done. Some patients are able to survive, but immunity after a disease is not found because of the amount of botulinum toxin is very small to cause the death of patients and not enough for immune system to recognize like antigen. Very small amount of botulinum toxin is enough to kill a patient, but not enough for immune system to recognize like antigen and develop the full immune response. So immunity is not developed. And if patient did recover all botulism one time, again, he is able to be infected by this botulinum toxin again. He has no immunity anyway. So now, in case of such symptoms, microbiological diagnostics should be done. And memorize, please, in case of toxinemic infections, the keyword is exotoxin. So keep in mind, the main virulence factor is exotoxin. Clinical symptoms are developed due to exotoxin action, specific symptoms. Then in microbiological diagnostics, the purpose of study is detection of exotoxin, and exotoxin should be detected as soon as possible. And just by detection of exotoxin, it is possible to confirm the diagnosis, and treatment should be done as soon as possible to save the life of the patient. So, detection of exotoxin, detection and identification of toxin by biological and serological methods, and isolation of pathogen by the bacteriological method, just for any case, okay? So, because the first task is to detect, to identify the toxin, what group does it belong to? Specimen, food, leftovers, the rest of food, if it is, and material obtained from the patient. So, patients have no vomiting, so sometimes it is necessary to wash the stomach, gastric lavage, blood, urine, feces, and maybe to the rest of food, I told you. Biological method for detecting botulinum toxin. The detection of botulinum toxin is carried out in a biological sample in laboratory animals using neutralization test. It is named biological method because of animals are used, but really we have to do a serological test, neutralization. The toxin should be neutralized, and it is possible to neutralize of toxin just by action of antibodies for exotoxin. named antitoxins. Sorry. Two groups of animals are used, control and experienced. And the animals of control group are injected with the filtrate of the test material, maybe feces, maybe rest of food, in which the presence of exotoxin is assumed. To carry out the neutralization reaction, a mixture of the test material with diagnostic antitoxic sera of different types is administered to the animals of the experimental group, A, B, E, F. This antitoxic sera produced and read not just A, B, A, F, all 8 types of antitoxic sera produced. I'm going to remind you, what does it mean, serum? If preparation is named serum, it contains antibodies for 13 for 13 species of microbes. In this case, we are talking about antibodies for exotoxins, monovalent sera, where antibodies are for 13 types of exotoxins. And so the control group will get just material where maybe exotoxin is, and other animals will get the mixture of the material plus antitoxic serum separately. And by the death of animals in the control group, the presence of exotoxin is possible to detect. But we have to know the type of toxin. The death of laboratory animal is not proving that the death was caused exactly by botulotoxin. And so we have to watch animals from experimental group. If they will remain alive in the event that neutralization of exotoxin with the corresponding antitoxic serum occurs. In this way, the type of toxin can be determined. I will show you picture. So neutralization test. So, and this is control, and this, and of course, it's not just one animal, maybe a few animals, and the material should be injected into this animal, and we see mice died. And then other animals should get mixture with material plus antitoxic serum. This group will get mixture with antitoxic serum type A. These animals will get mixture of material plus antitoxic serum B. And these animals will get injection of mixture of specimen, material, and antitoxic serum type E. Really, then maybe E, F, G, and so on, all of them. And now we have to see control group animals. No, mouse died. Now this mouse died, this mouse died, and this mouse, happy mouse, survived. So we should realize what antitoxic serum was used. It was antitoxic serum A, type A. So if these animals still survive, still alive, what does it mean? We did neutralize exotoxin by using of antibodies. It was antitoxic serum type A, means in the specimen, in the patient's body, exotoxin A is circulated. Why it is important? Because of for treatment of botulism, the first task is to neutralize exotoxin, and it is possible to neutralize exotoxin just by using of antitoxic serum, the proper type. So before we will detect what type of botulotoxin is circulated, the mixture of this antitoxic sera are used, the mixture, including A, B, E, F sera. But after we will detect exactly what type of exotoxin is in this material, the treatment of patient is done by using of monovalent antitoxic serum, the certain type. Maybe A, maybe B, maybe in this case, we are talking about type A. Do you see? So it's life important for patient to detect what type of botulinum toxin is in the patient body. It's not so simple like on this picture. There are many difficulties in biological method. Anyway, it is suggested. And it is better to use serological method, of course. Serological method. At the early stage of botulism, it is possible to detect botulinum toxin in the blood of patients. Because do you remember pathogenesis of botulism? It goes into the digestive tract, but then it goes to the bloodstream. And it is easy to detect botulinum toxin in blood by using indirect hemagglutination test. Specific for hemagglutination test, red blood cells are used. And in this case, antibodies for exotoxin are absorbed on red blood cells, and it's also to detect them. And ELISA test now is developed for detection of toxin and precipitation test. So any kind of immune test, it is possible to use for detection of botulinum toxin in the blood. There is this picture. This is passive hemagglutination test, and it is possible to detect botulinum toxin in the blood. In the blood, I'm repeating. Cultural method is not important, but it is possible to isolate these bacteria on RCM medium. And after cultivation on RCM broth, bacteria should be re-inoculated on blood sugar agar. And you should remind methods of cultivation of strict anaerobic bacteria. These petri dishes should be placed in a Macintosh and fill jar where the air is eliminated completely, is replaced by inert gas. And then after cultivation at thermostat, from suspicious colony are examined by microscopical method, and then suspicious colony is re-inoculated again on RCM broth. I told you about stages of isolation of pure culture of strict anaerobic bacteria, you know them. So all Clostridia, if you are going to isolate Clostridia, we should you should follow these steps: RCM broth, then blood sugar agar, and then again RCM broth for pure culture isolation. Treatment. Of course, patients are admitted at hospital, very severe cases. And for the treatment of patient, I'm repeating again, the first task is to neutralize exotoxin. Antibiotics are not able to neutralize exotoxin. Antibiotics are used for any case, maybe they are live bacteria. Metronidazole is used, for example. Clostridia are sensitive to metronidazole. And for the treatment of a patient, anti-botulinum HOS antitoxic serum is used. In our country, it is HOS. HOSes are available, so antitoxic serum against tetanus, against botulism, and against diphtheria are heterological sera. They are prepared by immunization, hyperimmunization of HOSes. And HOSes antitoxic serum is used for neutralization of botulinum toxin. Prior to establishing the type of a toxin, a polyvalent antiserum is used that contains antibodies to toxins of all types. A, B, E, those are very often isolated from patients. After laboratory detection of the type of the toxin, specific monovalent serum is used. There are photos of preparations. Antitoxic serum type A, it is HOSes. So before application of the HOSes serum, doesn't matter, tetanus, against diphtheria, against botulism, we have to, the allergic test, skin allergic test should be done. And this is serum diluted 1 to 100, especially for this skin allergic test, because realize that we are talking about HOSes serum. And even there are antibodies, they are HOSes proteins, and there are in the serum, there are another proteins, they are HOSes proteins. And if it's not the first time injection of the HOSes serum, patient will develop allergic reaction. And so it is dangerous. So we have to do skin allergic test to realize if this person has allergic reaction to the hosis serum. If he has an allergic reaction, but he is a patient with botulism anyway, the serum should be used. It should be injected in small amount, in small doses with intervals, and plus we have to give other medicines to prevent allergic reactions. So very carefully, because it is hosis serum or blood. Hosis are available, so we have to use them. This is serum type B. This is serum type E for prevention. Of course, it is better to use human beings immunoglobulin, and if humans are donors are available, sometimes human beings immunoglobulin is used. Hosis are available. Prophylaxis. For the specific prevention of botulism, botulinum vaccines are used, and exactly they are toxoids. Toxoids of types A, B, and E, they are produced in Russia. Toxoids, I'm going to remind you. So this is keywords for toxinemic infections. The main pathogenic factor is exotoxin. In pathogenesis of infection, exotoxin action. And then in diagnosis, exotoxin should be developed. For prevention, antitoxic serum should be used, or immunoglobulin. Immunoglobulin, again, antitoxic. And for prophylaxis, toxoids are used. Staphylococcal toxoid, diphtheria toxoid, tetanus toxoid, gas gangrene, there are 2 toxoids, and 3 toxoids for prophylaxis of botulism. And nowadays, for prevention of whooping cough, toxoid is used. So no live vaccines. In case of disease caused by Clostridia, no live vaccines, no killed vaccine, toxoids only. You did study toxoids. I'm going to remind you what is it, toxoid, because maybe some of you on exam will get preparations, and you will see, not serum, toxoids. You will get preparation named toxoid. So you should explain what is it. Toxoid, anotoxinum in Latin, is obtained from exotoxin of bacteria by treatment with a 0.4% formaldehyde solution at a temperature of 37 degrees by Celsius within 3-4 weeks. So what is the aim is exotoxin is a toxic protein, and not possible to use toxic protein for vaccination, for immunization. We will kill this person. So the task is to keep antigenic structure of this protein. We are not going to destroy it, but we have to keep antigenic structure, but we have to remove toxic properties. Ramon was the first scientist who did develop method, and he did use 2 factors. One is chemical factor, formaldehyde solution. Another factor is physical factor, temperature and time. So, and the toxin loses its toxic properties, but retains antigenic and immunogenic properties. During this time, during these weeks, formaldehyde blocks toxic centers of this protein, and we will get the same protein with the same antigenic structure, but without toxic properties. It has very mild toxic properties, and no side effects really is developed, but sometimes they are developed. So, and so what does it mean, immunogenic properties? So the protein is not destroyed, and it has ability to cause the production of antibodies when it is injected in the patient's body, in the healthy people. Healthy people should be vaccinated. So exotoxin is a toxic protein. Toxoid or anotoxin is the same protein without toxic properties. And toxoids, anotoxinum, are used for vaccination against toxinemic infections. Polyvalent. So people, absolutely healthy people, should be vaccinated. And people, those belong to risk group, are vaccinated in our country. And that people are soldiers in army because of they eat canned food and they are able to get wounds. So they are vaccinated by using of toxoids of all Clostridias, prepared of toxoids of all Clostridias. So completely this preparation is named sexta anatoxin. Sexta means 6. I told you 1 tetanus anatoxinum toxoid, 2 against gas gangrene, and 3 toxoids prepared of the exotoxins of Clostridium botulinum, A, B, completely 6. And sexta anatoxinum is used for prophylaxis of disease caused by Clostridia in Russian army. But sometimes after, so healthy people are vaccinated for any case, as usual. Vaccination is used for prophylaxis, healthy people. But sometimes if some people did eat the same product, and some of them did develop symptoms of botulism, it is late to use toxoids for specific prevention. And people who did eat the same food should get polyvalent antitoxic serum for emergency botulism prevention. It is not as usual prevention, it is emergency prevention. They should get the same preparation like people with symptoms of botulism, okay? Because it is too late to vaccinate them. What about non-specific prevention? Non-specific prevention in the production of canned products, meat, vegetable, fish products, it is necessarily to observe the sanitary and hygienic conditions for the sterilization of canned food and their storage. They should be kept at refrigerator because of cold, low temperature inhibit the multiplication of Clostridias. And sometimes canned food content of acid, any kind of acids, it's not good, of course, for people, but if pH is acid, the Clostridia are not able to multiply a lot. And at our country, it is suggested to people at home, do not close canned food tightly. It is possible to prepare, not possible to prohibit it people. It is not prohibited to prepare canned food, but the advice is don't close tightly by metal stoppers, just by plastic covers. And if the canned food is closed by plastic covers, there are no anaerobic conditions. But in this case, these goods should be stored at low temperature at refrigerator or any place with low temperature. So have you any questions about Clostridium botulinum, about botulism? Is it clear?
I think everything's clear, Dr. Thanks.
No questions. So the next lecture will be on 5th of November. I'm not sure. It is day for lectures. I will inform you, okay? So in 2 weeks from now, it should be on 5th of November. And then I will inform you. Are there any changes in our life or not, okay? We don't know. We are waiting for the order of the head of university. Sure?
Okay.
I will inform you. So if you have no questions, I will finish the lecture.
Okay, Doctor, thanks a lot.
Thank
Have a
you.
nice-
Thank you, doctor. Have a nice evening. Goodbye.
You're welcome. Bye-bye.
Have a nice day.
You too.
Bye-bye, Doctor. Have a nice day.
You too.